Hey guys, how are you doing? I hope you're doing well. I have prepared this short video for you in order to show you how to set up your workspace in order to run an SDS-PAGE for proteins. We're going to be doing this next week, so I prepared this short video in order to illustrate step by step the procedure that we need to follow in order to accomplish this practice. I hope you find the video useful. If there are parts of this video that are not clear enough, feel free to look for others in YouTube. I'm sure that there is videos that are way better than this one. This is my first time doing this, guys, so I apologize if it is not clear enough, but I hope you find it illustrative. So let's begin now. An important note: the models I am going to show you are different from the ones you are going to use during the practice, but you will get the idea. As usual, we will need a regular set of pipettes, a dry bath with a heating block to accommodate small microcentrifuge tubes. Make sure to set up the temperature of the bath at around 95 Celsius degrees. A vortex to properly mix our samples. A power source capable of delivering at least 200 volts. Again, specific brands and products are slightly different from the ones you will use, but bear with me. Milky water to prepare our buffers and if necessary dilute our samples. I like to use pre-chilled water, but if it is at room temperature, it is okay too. A concentrated stock of the running buffer. It is important to check we are using the proper combination of buffer and gel. There are different types of gels that can help us to achieve separation of proteins based on different principles. Today, I will use the second one, a regular 3 SDS gel, which is the most common format and is also compatible with the buffer I showed you before. Of course, you will need to have your properly labeled samples, as well as a sample buffer and a reducing agent, so you can break down the disulfide bonds in the proteins. Samples can be prepared with or without reducing agents according to your needs. A pre-labeled molecular weight marker would let you follow the separation process, and it is also very important to include one for the analysis of the gel that we will do at the end of the electrophoresis. And finally, you will also need a detection method for the proteins once we got them separated. In this case, I will use Kumasi G250, but there are many other options available. Prepare your working running buffer by diluting your concentrate in pre-chilled water. In my case, I took 100 ml of a 10x concentrate and diluted with 900 ml of water to get 1 liter, which is more than enough to run 1 or 2 gels. Ideally, keep the buffer in the fridge until needed. Oh, and don't forget to label your buffer properly. Next, mix the sample buffer and the reducing agent according to the instructions in the manual. This mixture will be used to dilute your samples, so you need to prepare enough for all your samples and a little extra just in case. We will prepare the samples in 0.6 milliliter microcentrifuge tubes, but before doing so, it is important to properly label each one of the tubes. I usually just write down a number and match that number with the identity of my sample in an Excel spreadsheet. To prepare your samples, you have to consider which type of sample buffer do you have. In my case, I have a 4x concentrate, so I need to mix 1 part of sample with 3 parts of buffer in order to achieve a final 1x concentration. So first, I would add 10 microliters of sample buffer to each tube. Just to show you what is the effect of the reducing agent, I also prepared a second set of samples without it. As before, I added 10 microliters of this non-reducing sample buffer to each tube. To complete the preparation, briefly vortex your samples. Take 30 microliters and place them in each tube that you prepared with sample buffer. When you are done with sample preparation, take the tubes with your samples and place them into the dry bath. Set up your timer, your watch or your cell phone for 5 minutes and let the time run. Okay, now it's time to deal with the gel. First, take the gel out from its package. Second, carefully remove the thin piece of green plastic located at the bottom. It is very important that you do not forget to do this. Third, carefully remove the big green piece that is located at the top of the gel. This piece is called the comb and is used to generate the wells in the gel when it is being prepared. Next step is to position the gel into the holder. Take the holder out of the tank and loose the latches. Place the gel into the holder with the tallest part facing toward you. Position the damp at the back of the holder and place the whole unit back and adjust the latches. Now, fill the space between the gel and the damp with 1x running buffer. The level of the buffer should be above the wells. Put the holder back into the tank and fill the tank with buffer all the way up to the middle. Now, it is time to load each well. First, place 7 microliters of the molecular weight marker into the first well. For the samples, we will use 15 microliters per well. It is convenient to use pipette tips with narrow ends and place the end of the tip all the way down into the wells. Take note of the location of each sample so you can create a reference list. Observe how the gel holder is color-coded to indicate the polarity of the two terminals. Place the lid on top of the tank matching the color of the terminals in the holders with the colors in the lid. Plug the cables into the power source, again respecting the color code. After you are done connecting everything in place, it is time to program the power source. I like to run gels using a two-step program. The first step is 10 minutes at 100 volts and the second step is 45 minutes at 200 volts. Now press the run button. If you observe the gel while it is running, you will see how the dye that is part of the sample buffer moves across the gel and gets separated into its two components. This is the running front, and most proteins are located above this line, unless they are very small. If you pay attention, you will see that the proteins in the lane loaded with the molecular weight marker got separated over time. Once electrophoresis is done, we need to detect the proteins in the gel. To this end, first we need to take the gel out of the tank and proceed to open the cassette. Locate the indentation in the edge of the cassette and insert the little green tool. Apply force and crack one side of the cassette open. Then, move to the other side of the gel and repeat the operation again. Both plastic parts should get completely separated in order to liberate the gel. Carefully remove the gel from the plastic cover and place it into a container where you will add between 50 and 100 ml of your staining reagent. Incubate for 1 hour under mild agitation. After an hour, we can begin destaining the gel. In this case, you can see how after an hour the color of the staining solution has changed. I will discard the blue solution and add 50 to 100 ml of water to the container and destain for approximately 1 hour. Now it is time to generate a digital image of our gel. I'm going to be using the Chemidog XR station, which uses a CCD camera to generate high quality images. After opening the tray, we need to place the white light conversion screen on top of the UV transillumination unit to have an appropriate type of lighting. Take the gel out of the container and place it in the center of the white surface. Close the tray and go to the computer. Find the Image Lab icon and open the software. We will select New Single Channel and then the type of application, which in this case is proteins and the Kumasi blue. By making click on Position Gel, you will have a real-time image of the gel in the tray, so you can properly center it in the middle of the screen. You can also zoom in or out to focus in specific parts of the gel or get a complete picture of it. Finally, press Run Protocol. The software will automatically adjust the exposure to get a nice contrast between the bands and the background. Save your image, and now you can proceed to analyze, modify, or print as needed. Of course, you can always take a picture with your phone and get something like this, so it is up to you.